Today we're going to be comparing the research article "The Conserved Structure of Plant Telomerase RNA Provides the Missing Link for an Evolutionary Pathway from Ciliates to Humans" to the CNN News article "Breakthrough Discovery in Plants' DNA May Lead to Slowing the Aging Process in Humans." This is a presentation by me, Abhigyan Shukla, for Arizona State University's Spring 2021 Bio 282 Lab Final. A little bit of background on the topic. Telomeres are basically protective caps for chromosomes, noted in blue on the red chromosome, and they're basically a random sequence of non-coding DNA, in this case GGTTAG is the sequence, that is used to stop the actual chromosomal DNA from being damaged by exonuclease. Every time the cell divides, specifically in the germline cells, you see exonuclease take a little bit off of telomeres every single time per cell division. So these excised portions, while not important, protect the chromosome from being excised. And telomerase, which is the actual enzyme, effectively just extends these telomeres so that chromosome isn't damaged. The chromosome, once the telomeres expire, is going to be excised by exonuclease, and that leads to a plethora of the problems that we see in aging, such as an inability to heal quickly or a greater proclivity to incurring cancer because your chromosomes are actively being damaged. So within the actual telomerase itself, there's a complex. So you have TERT, which is the protein in the complex, and you have the RNA template. Because TERT is common across all organisms, the only part that changes is the RNA template, which is the focus of this study. For the background and goals, this was conducted by researchers at both ASU and Texas A&M in collaboration. And they were looking to find the RNA component of telomerase in plants, specifically in Arabidopsis thaliana, which is a flowering plant, and it has a common genome with a multitude of other flowering plants and plants across the world. And we have to make a clear distinction here. Telomerase is an RNA-dependent DNA polymerase, which asks us to look at RNA and DNA, while the central dogma of genetics says that you go from DNA to RNA to a protein. In this case, it's a little bit different. You go from an RNA template to DNA, which is added onto the end of a chromosome. And so basically what this study was seeking to do was bridge the gap to get a greater evolutionary understanding of telomerase. In 2009, we were able to get an understanding of ciliates telomerase in the RNA template in there, and further from that, we were able to get one for humans. But currently, the version for plants was missing until this study. We weren't sure how to sort of bridge the gap and what the evolutionary impacts of telomerase were. So the procedure to actually conduct the study was done using RNA immunoprecipitation, or RIP for short. The way that this occurs is you first lyse the cell, either using a chemical component which destabilizes the membrane or splicing it manually. This allows for the extraction of the proteins and RNA from within the nucleus of the cell, and then antibodies are bound to that. Since TERT is the protein in the protein complex of telomerase, what you do is you take anti-ATTRT, which is the antibody, and bind it to that protein. That allows for the RNA to be isolated, which can then be amplified or replicated using polymerase and in primer sequences, and then PCR to see the actual bands in specific nucleotide sequences. The results are pretty profound. What was found is that ATTR, which is the RNA template found in this study, is the predominant RNA associated with active telomerase in the Arabidopsis population, meaning that this RNA template can be combined with TERT, which is the protein complex right here, and you can create telomerase outside of the in vivo population, meaning you can create it in vitro or outside of the organism, and it is just as effective in either place, meaning that this RNA template can be used to create the actual telomerase molecule or enzyme outside of the actual organism to be effective in other places and to also be studied in conditions. But moreover, what was found was that the plant RNA sequences are actually homologous with human ones, which means that they stem from a common ancestor. That's very important because if we're able to sort of track the lineage and evolution from ciliates to plants and then eventually to humans, we may be able to grasp a better understanding of telomerase and genetic diseases that are related to it. In the figure itself, we see a phylogenetic comparison of those RNA components, and specifically RNA pseudoknots. While researchers aren't sure what exactly they do in the RNA template, we do know that there's sort of a triple helixed portion of RNA that folds in back on top of itself in order to sort of stabilize the template. And so as you can see in the highlighted populations, which are ciliates, plants, and mammals, there's an increase in complexity over time in the phylogenetic comparison. While ciliates tend to only have one RNA pseudoknot, plants can have two, and mammals such as humans can have 3 to 4. While we're not sure exactly what that complexity leads to, it does open a number of doors for analysis. But moreover, what we find is that in plant populations, because those RNA templates are so similar, most plants are actually going to be following this sort of 2 RNA pseudoknot structure, and you can extrapolate this to create the actual telomerase in other plant populations. The article does a very good job of sort of expressing this sentiment. So, outside of a occasional few misnomers, such as the title, which says "Breakthrough Discovery in Plants' DNA," as the actual article or research paper was looking at the RNA template in the telomerase, and the idea or extrapolated impact of leading to the slowing aging process in humans, which, while briefly touched upon in the actual study itself, is not the main focus, as the study focuses on more of the evolutionary impact and being able to trace what the mutations and evolutions were and why they occurred. The study actually does a very good job of breaking it down in layman's terms. It really focuses on what telomeres are, how they impact chromosomes, and how they protect humans from being able to get into diseases. And more specifically, it also breaks down the potential impacts of plant research on human-based life forms, such as how we could use the telomere information we get from this study on trying to stop cancers or potentially even stopping aging, like it says. So overall, it does a pretty good job. And here are the credits. All of the actual drawings were made by me.